In this explainer, we're going to analyze a 2025 paper by Nicholson and colleagues. This work puts forward a compelling argument that challenges our current understanding of a group of severe neurodegenerative disorders. Specifically, it proposes a significant shift in how we think about the GM2 gangliosidosis, a shift that moves our focus from a problem deep within the cell to a rather unexpected set of consequences happening right on the neuron's surface.
So, here's our roadmap. First, we'll cover the fundamental ganglioside catabolism problem. Then, we'll look at the in vitro model they developed to study neurodegeneration. After that, we'll examine the proteomic investigation and what it revealed about the remodeled plasma membrane. From there, we'll connect these molecular changes to neuronal hyperactivity. And finally, we'll discuss the new disease paradigm this research suggests.
All right, let's begin. To really grasp the core problem, we first need to review some fundamentals of glycosphingolipid metabolism. You'll see that the central issue in diseases like Tay-Sachs and Sandhoff is a very specific failure, a breakdown in a critical enzymatic pathway located inside the cell's main recycling hub, the lysosome.
So, what exactly are these molecules? Glycosphingolipids, or GSLs, are bioactive lipid components that are integral to the outer leaflet of the plasma membrane. A particular subtype of GSLs, known as gangliosides, are found in high abundance in the brain. In this context, they're crucial for modulating membrane properties and participating in various cell signaling cascades. This brings us to the GM2 gangliosidosis. This is a class of inherited neurodegenerative disorders that emerge when the catabolism of a specific ganglioside, GM2, is impaired. The consequence is a progressive and toxic accumulation of this lipid, primarily within the lysosomes of neurons. And it's this accumulation that drives the severe pathology we see in conditions like Tay-Sachs and Sandhoff.
The degradation of GM2 is a very precise multi-step process. It begins when an activator protein, GM2AP, presents the GM2 ganglioside to the enzyme β-hexosaminidase A, which we'll call HEXA. HEXA's job is to cleave a terminal sugar from GM2, thereby converting it into the ganglioside GM3. Now, the problem arises from mutations. Tay-Sachs disease is caused by mutations in the HEXA gene, which codes for the alpha subunit of the HEXA enzyme. Sandhoff disease, on the other hand, results from mutations in the HEXB gene for the beta subunit. But in either case, the outcome is effectively the same. Functional HEXA is absent, the pathway is halted, and GM2 begins to accumulate.
Now, a significant challenge in studying these diseases has always been the development of relevant models. Standard animal models, for instance mice, often have different ganglioside metabolisms compared to humans, which limits their utility. The work we're looking at today addresses this challenge directly by employing an advanced human cell-based platform.
The system they used is called the i3 neuron, or i3N platform. This technology starts with an induced pluripotent stem cell line that's been engineered to contain an inducible GM2 transcription factor. The key advantage here is that this system allows for the rapid and reproducible differentiation of these iPSCs into a highly homogeneous population of human cortical neurons. This, of course, provides an excellent isogenic substrate for their subsequent molecular analyses. So, to model Tay-Sachs and Sandhoff disease in this platform, the investigators employed CRISPR interference, that's CRISPR-I, to suppress the expression of the target genes HEXA and HEXB. And the efficacy of this approach was confirmed using quantitative PCR. The data showed a highly efficient knockdown, with the target mRNA levels being reduced to just 0.5 to 3% of the levels seen in the control cell line. Of course, a reduction in gene expression is one thing, but the functional consequence is what matters. And indeed, subsequent enzyme activity assays confirmed a corresponding profound loss of HEXA activity in these knockdown cell lines. It's worth noting that the low level of residual activity they observed is consistent with the levels found in patients who have the most severe, infantile onset forms of these diseases.
A crucial step, then, was to validate that these engineered neurons accurately recapitulated the key cellular hallmarks of the disease, and multiple lines of evidence confirmed that they did. For example, immunofluorescence imaging showed GM2 accumulation within LAMP1 positive lysosomes. Electron microscopy revealed enlarged endolysosomes that contain those characteristic membranous substructures, the whorls and zebra bodies. And finally, glycan profiling confirmed a greater than 20-fold accumulation of GM2, with no other gangliosides being significantly altered. So taken together, these findings really validated the i3N system as a high-fidelity disease model.
Okay, so with a validated in vitro model in hand,
The next phase of the research was to perform an unbiased, large-scale analysis. The goal was to understand how the accumulation of just a single lipid species could impact the entire neuronal proteome.
This really gets to the central question of the study. What are the global, far-reaching consequences of this one single metabolic blockade? We've known for a long time that GM2 storage is the primary defect, but its downstream effects on the neuron's broader protein landscape, well, that has not been very well defined.
The whole-cell proteomics analysis yielded some very interesting results. It revealed a significant and quite specific upregulation of proteins that are associated with the endolysosomal system. This included a range of proteins, degradative enzymes, lipid transporters like NPC1 and SCRB2, and also proteins involved in membrane trafficking and exosome formation, such as the tetraspanins. This overall pattern is strongly suggestive of a cellular response that's focused on handling this increased lipid burden, essentially an attempt to clear the accumulating material.
This specific enrichment of trafficking and exosomal markers pointed toward a particular cellular clearance pathway known as lysosomal exocytosis. This is a process where the lysosome itself fuses with the plasma membrane to expel its contents into the extracellular space. Now, while this can serve as a mechanism to clear out cellular debris, it also comes with a potential consequence. It could alter the protein and lipid composition of the cell surface.
So this proteomic evidence for an uptick in lysosomal trafficking and exocytosis led the researchers to a new hypothesis. They proposed that these intracellular events were not just a clearance mechanism, but that they were actively remodeling the composition of the cell's external boundary, the plasma membrane.
To test this new hypothesis, the team then performed plasma membrane proteomics. The analysis did, in fact, confirm their prediction. It revealed the inappropriate appearance of lysosomal membrane proteins, things like SCRB2 and TSPN3, on the cell surface. But just as importantly, the data also showed significant alterations in the abundance of a number of key synaptic proteins. This included, for instance, an increased abundance of the calcium sensors Synaptotagmin-1 or SYT1, and certain neurotrophin receptors, along with decreases in other proteins that are critical for normal synaptic function.
So if we compare the two proteomic datasets, a cohesive narrative begins to form. The whole-cell proteomics revealed a systemic stress on the endolysosomal pathway. This, in turn, appears to trigger a compensatory clearance mechanism through exocytosis. Then, the plasma membrane proteomics shows us the direct result of that mechanism, a remodeled cell surface, one that is not only decorated with mislocalized lysosomal proteins, but also possesses a functionally distinct composition of synaptic proteins.
Documenting these proteomic shifts is, of course, a significant finding in itself. But the critical question that follows is a functional one. Do these molecular alterations actually impact a neuron's primary function, that is, its electrical activity?
To address this question, the researchers used multi-electrode arrays, or MEAs, to measure neuronal network activity. The results were quite clear. The disease model neurons exhibited hyperactivity. When compared to the control neurons, they fired significantly more spikes per network burst, and the duration of these bursts was also significantly longer. Together, these observations indicate a state of increased network excitability.
There was another interesting finding. The two cell types responded very differently to external electrical stimulation. The control neurons largely continued their spontaneous firing, seemingly unaffected by the stimulus. The disease model neurons, in contrast, began to fire in synchrony with that external stimulation. This result demonstrates that the disease model neurons have a reduced threshold for depolarization, which makes them hyperexcitable. This is a functional change that is highly consistent with the proteomic alterations they observed, particularly the increase in proteins like the calcium sensor SYT1.
The final piece of the investigation asks whether these findings represent a mechanism that is conserved across related disorders. And the answer to this question leads the authors to propose what is essentially a new paradigm for this entire class of diseases. To see if these mechanisms were specific to GM2 storage, the team created another neuronal model, this time for GM1 gangliosidosis, by knocking down the GLB1 gene. They then performed plasma membrane proteomics on this new model, and the results revealed a very similar outcome, a significant enrichment of lysosomal proteins, including TSPN3 and SCRB2 at the cell surface. This finding strongly supports the conclusion that this process, lysosomal exocytosis, and the subsequent remodeling of the plasma membrane is a conserved pathogenic mechanism across the gangliosidosis. So based on this collective body of evidence, the authors conclude the traditional classification of these conditions as purely lysosomal storage diseases is in fact incomplete. They posit that the gangliosidosis should also be conceptualized as disorders of the plasma membrane, where these complex changes to the surface composition directly impinge on and disrupt neuronal function.
This reframing of a class of rare diseases naturally prompts a broader and perhaps more provocative question. We know that lysosomal dysfunction is a common feature in many more prevalent neurodegenerative conditions, such as Alzheimer's and Parkinson's disease. This research, therefore, suggests the possibility that this lysosome-to-plasma membrane access could represent a more general and perhaps underappreciated component of neurodegenerative pathology as a whole.